welcome address. I welcome you, ma'am.
So thank you, Kavya Rasim. So good afternoon to one and all present here. So I am happy to welcome you all for the 70th National Level Virtual FDP program on prospects and retrospects in life science. So this is the 4th day session. So we are running it successfully. We are happy for that. So we are also getting a lot of comments from different delegates from the universities and colleges appreciating the specialization of delegates that we have chosen. So thank you. It really energizes us to do more and more again. So to say about this one, it is a DPT sponsored FDP. So as RVS comes under DPT Star Scheme College, so this is a funded one, and we are doing this 7 days program entirely to enhance the skills and research part of the faculty members. And today, on behalf of the Department of Biochemistry, Ratnavale Subramaniam College of Arts and Science, on behalf of the management, principal, vice principal, and the DPT Star College coordinator, and all the faculty members, I welcome our eminent chief guest, Dr. T. Murali Krishnan, assistant professor from the Department of Biochemistry, Rajaji Sarvoji Government College, Tanjavur. So to say about him, he's a former HOD over here in RVS. So he's the one who blessed us and gave us the department when he left in 2007. So all these days, we are very trying hard to bring him for somehow to meet our students and faculty members, but hardly we couldn't do it. So this is the first time we are in intact contact with him. We are happy to have you, sir, over here. So we are very much eager to listen your topic, recent approaches in medical research. So welcome you, sir, to the arena.
Thank you, ma'am. Thank you so much. So now it's my pleasure to invite Ms. Lalita, Assistant Professor, Department of Biochemistry, to give a special introduction about our chief guest. I welcome you, ma'am.
Thank you, Kavya Rasim, ma'am. On behalf of management, dignitaries, Department of Biochemistry, it's my immense pleasure for me to introduce and welcome our chief guest, Dr. V. Murali Krishnan, Assistant Professor, Department of Biochemistry, Raja Sarfoji Government College, Tanjavur. I feel glad to inform you all that he served his career as HOD, Department of Biochemistry, RVS College during the period of 2003 to 7. Dr. Murali Krishnan completed UG Biochemistry from Nagamalai Navarisam College, Erode, PG and Doctorate in Biochemistry from PSG College of Arts and Science, Coimbatore. His area of interest is environmental biochemistry, bioprocess technology, cancer biotechnology, microbial biotechnology. He has guided 6 MPhil scholars. He has published more than 20 research articles in reputed national and international journals. He is a reviewer in Elsevier journals. He has also applied for a patent. He has organized and participated in national and international conferences, and he was honored by Best for Paper presentation also. His projects was internationally recognized by United Nations Environment Program Canada. His project agreement also signed with National Biodiversity Authority, Government of India. Highlighting his administrative position, he has worked as a HOD, Biochemistry, RVS College of Arts and Science, then HOD in Government Arts College, Paramakudi, then head in charge unaided college from PSG College of Arts and Science during the period of 2003 to 17. Apart from this, Dr. Murali Krishnan plays a dynamic role, acted as a district organizer, program officer, coordinator and member in Youth Cross, Red Ribbon Club, Government College, Paramakudi. Besides this, he is a BOS member in various colleges. He has a lifetime membership from Indian Science Congress, Society for Biochemists, India. Such an eminent personality is here to share knowledge with us. Once again, I welcome you, sir, on behalf of all.
Thank you. So may I start the session?
Yeah, without taking much time, we hand over the session to you, sir. It's your forum.
Yeah, so good afternoon, one and all present here. So first of all, I feel so elated and happy to be there, to be getting associated myself to RVS College of Arts and Science after 14 long years. And so at this time, I would like to thank Dr. Shamina for giving me an opportunity, as well as I would like to thank Dr. K. Senthil Ganesh sir, our managing trustee, as well as Dr. T. Shivakumar sir, principal, who was my colleague earlier, as well as Dr. Dhanapalan sir, microbiology, my friend, my good friend always, and Ayapadas, head of biotechnology. And I would like to fondly remember Dr. H. Newman, ma'am, who was the secretary earlier, as well as Dr. P. Jairam, sir, principal, earlier principal at RVS. And with these things, let me go into the session. And this session is about a topic which is very much fascinating me right from my school days. So let me go into this. Ma'am, is the screen visible?
Yeah, it's visible, sir. Visible.
Fine. So
Fine,
today's topic is recent approaches in medical research, which is a very big topic actually. So I would like to narrow myself to organoids. So my topic will be an introduction to organoids. And coming for the few recent approaches that are happening throughout the world. It starts from cell culture, developed to expand culture, and now going on is 3D organ culture. And organoids is an important part that comes under 3D organ culture. There are also various other topics like 3D bioprinting, nanorobotic surgery, which are gaining more importance nowadays. But then I would like to select organoids, which is always fascinating. And in this presentation, I would like to give an introduction on 3D tissue culture, reasons for selecting 3D culture, cell potency and organoids, its role in organoids, organoids, a definition, its history, properties, process, the types of organoids that have been formed throughout the world, applications of organoids and market scope. And before going into that, I would like to tell you that this is a topic which is gaining more importance in the past few years. I have to say it started from 2010 exactly. And currently it is a $500 million US dollar business. So let us start with the types of 3D tissue culture. Basically, there are three types of 3D tissue cultures. One is scaffold, which uses hydrogels or inert matrices where the cells can be cultured within a supporting scaffold to allow the growth in all directions. And second type is scaffold-free 3D cell culture, where without the presence of the supporting scaffold, the cells will be allowed to grow. And this includes low adhesion plates, micro-patterned surfaces, and hanging drop techniques. And third is microfluidic organ-on-a-chip models, which again is a very new trend in the medical research. So out of these, I would like to take a different thing, that is organoids, which is higher than that of a cell culture and bit lower than that of, or we are on a stepping stone towards the three-dimensional organ culture. Right. So why should we go for a 3D culture always? Why do the medical researchers are going behind three-dimensional organ cultures or three-dimensional cultures basically? The reasons are simple. Number one, they better mimic the tissue-like structures. Number two, it exhibits differentiated cellular function. And number three, it can co-culture two or more different cell types within a single medium, and it can stimulate the microenvironment conditions and can also control the nutrient gradients. And more than everything, these kind of systems can very well predict the in vivo responses to the drugs to which we are going to give. And very recently, scientists have found ways of culturing the organ-specific tissues from stem cells that could change the way diseases are studied and treated. And out of all these things, the organoids are playing. They have gained a elated position, and it's a new approach into the disease development discovery. Right. So next we will go to the second thing, that is the cell potency and the organoids. So as I told you already, organoids are nothing but higher than that of cell culture and lower than that of three-dimensional tissue culture. So these organoids basically starts from the cell and its potency. So cell's potency is the ability to differentiate into other cell types. And we know that it is a continuum. So there are many things like totipotency, pluripotency, multipotency, oligopotency, and unipotency, which we will be just brushing up as we have learned about it already. So totipotency basically is the ability of a single cell to divide and produce all of the differentiated cells. So the very well known example is zygotes. We all know the human developmental model. So where the single totipotent cell zygote is getting which is formed after fertilization is divided into identical totipotent cells, which again on further growth is converted into the embryonic stem cells, which is nothing but a pluripotent stem cell. Next is pluripotency. So pluripotency is a term that refers to a stem cell that has a potential to differentiate into any of the 3 germ layers. Say for example, if it is getting itself converted into an endoderm, it is going to be forming interior stomach lining, GI tracts as well as the lungs. When it is getting converted, differentiated into mesoderm, it will be forming muscles, bone, blood, urogenital organs. And when it is getting differentiated into ectoderm, it will be forming epidermal tissues as well as the nervous cells. So pluripotency is a very important term which is involved in the formation of organoids. And this cell pluripotency also has got a continuum because there are 2 types. Either it is completely pluripotent cell or incomplete or partially pluripotent cell. Apart from these 2, there is one more thing that is called as induced pluripotent stem cell. This again is very much useful for us in this organoid, which we'll be seeing in future, in further slides. So what is induced pluripotent stem cells? So these are nothing but, these are derived from non-pluripotent cell, which are typically an adult somatic cell, which can be given or which can be taken from a patient. And we are going to induce it by forced expression of certain genes and transcription factor and converting it into a pluripotent stem cell. So I repeat, it is an induced pluripotent stem cell. So to be simple, this is making this market more valuable because we can make pluripotent stem cells even from the patients who don't have or whose stem cells have not been saved. So that is the thing. And here epigenetics also plays an important role in this conversion or reprogramming. So to be simple, the somatic cells can be reprogrammed to form pluripotent stem cells, and that's what is called as induced pluripotent stem cells. And next is multipotency. This again is very simple. There's a set of cells which have the gene activation potential to differentiate into discrete cell types. Say for example, we have got blood cells which can differentiate into lymphocytes, monocytes, neutrophil, etc. But this is not going to make any other cells apart from blood cells. And again, we have got oligopotency. Oligopotency is nothing but, for example, lymphoid and myeloid stem cells, which can make B and T cells. Again, this oligopotency is another one important property which makes this topic very important because we are in need of therapeutic agents like T cells, B cells, and we can make specific or patient-specific cells from this kind of potency. And now last is unipotency, which is nothing but the concept which explains one stem cell has the capacity to differentiate into only one cell type. Say for example, hepatoblast, which can give you hepatocytes. Right. So basically it goes on like this. A sperm cell fuses with the oocyte by means of fertilization, enters into a morula stage where we get the totipotent cells, which again on further growth makes blastocysts, and this blastocyst will be having the inner mast cell. The inner mast cell will be having the pluripotent capacity, and by taking these pluripotent cells, we will be able to grow the individual organs which are unipotent in nature. So hope you understand the importance of potency of the cells. So whichever cell you are going to take for this experiment to make, it should be pluripotent in nature. So that is a basic thing. Right. Next, we will go to the definition of the organoid. So organoid basically is a miniatured and simplified version of an organ which is produced in vitro in 3 dimensions that shows the realistic microanatomy. So for doing a research in an organ, you don't need an animal, you don't need an animal model, you don't need an experimental in any kind of experimental animal, or you don't need even cells. But just by making an organoid, you will be able to get what an organ is going to do. So they are basically derived from one or few cells from a tissue or an embryonic stem cell or induced pluripotent stem cell, which can self-organize in three-dimensional culture owing to their self-renewal and differentiation capacities. I repeat, this is derived from either one cell or few cells from a tissue, from a patient, or embryonic stem cells or induced pluripotent stem cells, and this has got the capacity to self-organize itself into three-dimensional culture and making it into a mimicking organ. Right, so this is how it goes of basically either you select the cells from a blastocyst where you isolate the ICM and you get the ECESCs, and similarly from a patient or a donor we can go for reprogramming to get the pluripotent stem cells, and we get induced pluripotent stem cells or pluripotent stem cells or embryonic stem cells, and these can be differentiated further into ectoderm or endoderm or mesoderm. If it is getting differentiated into ectoderm, this can be further differentiated into retinal epithelium to make retina, or neuroepithelium to make brain organoids, or endoderm can be further differentiated into hindgut endoderm to make intestinal organoids. Foregut endoderm can be differentiated into liver organoids and stomach organoids, and mesoderm can be differentiated into kidney organoids. So to be simple, there is a big difference between organ and organoids. The organoid is a miniature version of an organ. Right. So now coming for the history of organoids. Though I said that it is the entire research of organoids are being happening, important, all the milestones are happening. The growth of this particular organoids is happening only for the past decade. Actually, it started in 1907. So a scientist named Henry Van Peters Wilson, he did an experiment with sponge, and he observed that mechanically dissociated sponge cells can reaggregate and self-organize to generate a whole organism. So he did a mechanical dissociation of the sponge cell and observed that it has got the capacity of reaggregating and self-organizing to generate back its own whole organism. So there is where the tissue culture as such started. So this is the timeline, and we will go into this. So in 1907, the work was done with the sponge cells. That is, dissociation reaggregation experiment with sponge cell was done. And again, in 1944, the same dissociation reaggregation experiment was done with amphibian cells. And similarly, a similar work in 1960 happened with embryonic chick, and in 1961, differentiation of embryoid bodies in vitro happened in 1961. And in 1981, a very important milestone in the tissue culture history is the first ever isolation of pluripotent stem cells was established from the mouse embryo. And in 1987, a group of scientists, they could isolate the breast epithelia and organize into 3D ducts and ductiles in ECM extract. What they could prove actually is, they could prove that by growing these breast epithelia in a laboratory, they could even make those breast epithelial cells to produce the milk protein, and that made the basis for today's very biggest market of making commercial products from these cell cultures. And in 1998, embryonic stem cell line established from human blastocysts. And again in 2006, iPSCs, that is induced pluripotent stem cells, were identified, isolated, and established for the first time in 2006. And from here, the organoid growth have started. And again in 2008, 3D cerebral cortex tissue from ESCs were produced. And again in 2009, gut organoids from adult intestinal stem cells were produced. In 2010, gastric organoids, it was developed. In 2011, retinal organoids from mouse cells, mouse embryonic stem cells was developed. And again in the following year, 2012, the same retinal organoids were developed from the human cells. And in 2013, we started working on cerebral organoids from human cells, and the kidney, liver, and pancreatic organoids were developed. In 2014, again, prostate, lung organoids were developed, and gastruloids were developed. In 2015, mammary glands, fallopian tube, hippocampal organoids were produced. And again in 2019, a very important milestone happened. It was not given here, but in 2019, a very important milestone of brain organoids, which was developed. They've started applying this brain organoids for studying and treating various neurodegenerative diseases. So this happened in 2019, and from 2019, the whole most of the companies they started working on it. And in 2020, recently, the snake venom gland organoids were also produced. So now coming for the properties of the organoids. So as I told you, I repeat, the organoids are higher than that of the cell culture and lower than that of, or it is a stepping stone for three-dimensional cell culture. So coming for the properties, the basically will be having multiple organ specific cell types. So though it grows as an individual, it has got the capacity of growing multiple organ specific cell types. And very important second point is it is capable of recapitulating some specific functions of the organ. That is, to be simple, though it is not a complete organ, it has got the capacity of showing certain properties. If not all the properties, it has got the capacity of showing certain properties of organs. Say, for example, contraction, neural activity, endocrine secretion, filtration, and excretion. So all these properties we can observe even in the organoids. And third is the cells are grouped together and spatially organized, and they function similar to that of an organ. So this particular topic, that is organoid, have got a very big very big application in the near future. Now coming for the process of production of these organoids, the organoid formation generally requires culturing the stem cells or progenitor cells in a 3D medium. So this is the basic thing. So we are going to grow the cells, that is embryonic stem cells, in a 3D medium. And the 3D medium may be an extracellular matrix hydrogel or anything that depends upon the system which we would like to grow. And the organoid bodies can thus be made into grown by means of embedding the stem cells in the 3D medium. So initially we have to select the stem cells. The cells may be the stem cells may be taken from the embryonic stem cells. It may be pluripotent stem cells, or else we can also collect the cells from the patients, and the patient's cell, that is somatic cell, can be converted into pluripotent stem cells by means of inducing. So that is what is induced pluripotent stem cells, and these cells will be embedded, okay, in the 3D medium. And when the pluripotent stem cells are used for the creation of the organoid, the cells are usually allowed to form the embryoid bodies. And these embryoid bodies are then pharmacologically treated with whatever drug you want to test. Say, for example, you have developed a drug and you want to test it in particular organ. So instead of doing the research in that animal or an organ, we can go for that particular organoid, and you can divide into two. And you can give your own drug of interest, whichever you are interested to work on, and then you can compare. You can compare these two. That is pharmacologically treated with the pharmacologically not treated, and this is going to be reliable. So that makes these organoids to be very important. Also, these organoids have been created to use adult stem cells. Right. So this is how it goes of basically the cell conditions, that is initial cell conditions, can be, or this process, the entire process can be started sometimes with a single cell, or else with a group of homogeneous cells, or also with heterogeneous group of cells. Say, for example, if you're going to start with a single cell, imagine you are going to start with a single cell. A single cell is grown and it is converted into a homogeneous cell population, which again is allowed for undergoing differentiation and asymmetry generation, that is by means of self-patterning and self-assembly, which again undergoes maturation and self-assembly to make self-morphogenesis. It undergoes some morphological changes and is converted into an organoid. So this is a basic thing. And sometimes you may not be starting, you need not always start from a single cell. Sometimes you may be taking a homogeneous cell. So homogeneous group of cells are being taken. Again, it is also allowed for asymmetry generation, and it gets differentiated and it is further undergoing other morphogenesis and forming an organoid. And a best example for a single cell usage is our intestinal organoid, and best example for a homogeneous group of cells is optic cup organoids in eyes. And similarly, there is also a third, that is, we can also start the same experiment by co-culturing of different cell types, that is mixing of more than one type of cell. And this kind of thing is happening in case of production of liver organoids. So whenever you are going to make a liver organoid, we will be selecting multiple number of cells. And coming for the types, so there are many different types of organoids that are being produced and are being what to say? They are being produced and they are being marketed throughout the world. It is being marketed actually. So out of all this, very important is brain organoids. So we are going to see that first. So brain organoids is nothing but it's a very important thing which is considered to be a boon for us, because the current knowledge of the human brain is mainly based upon the post-mortem corpse brain specimens. There is a thing called as brain research in BRI, that is Brain Research Institute in England, and a scientist before almost like 1.5 decades, he gave a very big lecture on the ethical issues they have on working with the the restrictions in using human models, because you can never compare a human brain with any other brain of animal or insect or whatever it is. You can compare any other organ, but you can never compare a brain, because human brain is totally complicated and it has got lots and lots of different properties. So using animal models is not a good option, but even then they are using it, and even if the animal models are used, it is not going to give the exact answer whatever we wished. So in these kind of deficiencies, this kind of brain organoid development have made it possible to study, that is to grow a human brain inside a laboratory, and we can study that. So this is also called as the cerebral organoid or brain organoid. The cerebral organoid describes artificially grown in vitro miniature organ resembling the brain. So this is created by culturing the human pluripotent stem cells in a 3-dimensional rotational bioreactor and develop over a course of months. Okay. And why do we need a course of months? To be simple, it can be grown just even within days, but growing for a longer term has been worked out with few researchers, and what they have identified is when a brain is grown for a course of months, the development of the brain and the properties will be very high, so that we can study more number of properties. Right. And the procedure has the potential applications in the study of both the physiology of the brain as well as brain functions. And what is more important in this brain organoid is the cerebral organoids can experience even simple sensations in response to external stimulation. Say, for example, if you want to check, say, for example, you're doing a research in which you would like to check how a brain responds for particular smell. Okay. Say for example, you are surrounded with one particular smell, good or bad, and your brain starts responding to it immediately. So if you want to record this particular thing, at present there is no proper methodologies to do this. Currently we are using insect models for that, but we can never tell an insect model, we can never explain the human brain function with an insect model. So in this case, by means of cerebral organoid, even a simple sensation will be able to record, it will be recorded in the cerebral organoid. Right, so this is how it is grown. So human pluripotent stem cells are taken and they are cultivated to form the embryoid bodies, and it is induced to form the neuroectoderm, and it is grown in the gel droplet, and then it is spinned in the bioreactor, that is the cerebral organoids are grown in the bioreactor, and then it is allowed to grow for a longer period of time to get a complete organoid which can be used for various applications. So this is what is actually happening when you're going to compare the fetus brain development to that of the brain development in your laboratory. So you can find here it happens in the months in case of fetus brain development, where it takes approximately 6 months to get a mature brain in fetus. Whereas in your laboratory, by taking an induced pluripotent stem cell colony, see, you can find within 7 days, we can find the embryo body. Within 11 days, we can find neuroectoderm. And in 14th day, we will be able to identify, we will be able to make a neural epithelium. And in 20 days, we will be having a cerebral organoid, which is of a small piece. And this cerebral organoid will be transferred to the bioreactor, allowed it to grow for a longer period of time to get a bigger or an organ similar to that of brain. So it is possible in case of laboratory. So just imagine, it's like growing a brain in a lab and visualizing it daily is something, it's something so fascinating. We cannot do that at all. Right, again. So what is the use of this brain organoids? Number one, we can understand the development of brain, aging of brain, and evaluation of the human brain. So all these things are becoming very easy now. I told you in 2019, they have started working more sincerely on this particular topic, and many things came out, particularly treatment of various diseases, brain-related, brain regenerative or degenerative diseases are being processed in 2019. And up to today, brain organoids have been used in exploring mechanisms for neurological diseases and also drug screening. So even now, screening of drugs and its influence on the brain is a very important parameter to cross the drug system. So on the basis of the stem cell technology, the emergence of the 3D organoids have attracted great attention in the regenerative medicine. So in case of regenerative medicine, by means of this kind of organoid techniques, we will be able to reproduce even specific brain structures. Say, for example, if someone's brain cells have got damaged in only one particular area, it is possible for us to create that particular region alone, specific for that particular patient, and this can be transplanted into that person. So all the possibilities are there here. And currently, making of these individual brain regions have become possible now. And thus the brain organoids have become the excellent model for investigating the brain development as well as mechanisms of the related diseases. Okay, right. So this is the way it happens. So basically, you can either select ESCs or embryonic stem cells, or else you can also select somatic cells from a patient. Okay. Say for example, we don't have embryonic stem cells, then we can, for a mature patient, we can select either a urinary cell or fibroblast or dental pulp cell or monocyte, and by means of reprogramming, we can convert it into PSC, that is iPSC, induced pluripotent stem cell. And either by means of ESC or iPSC, it can be converted into an embryoid body, and then it can be developed into a brain organoid, which is used, right? So what are the future focus about the brain organoids is? Number one is prolonged culture time. So what scientists have observed recently is, by growing this brain organoids for a longer time, we will be able to identify various functions. We'll be able to get the organoids showing many functions of the brain. So that is the first thing. Number two, by means of sliced culture, we will be able to observe the physiology of the growth of the brain. Number three, again, we can go for a culture on a chip technology, which again is on a growing verge. And last is, this again is it's an expected advancement. That is, by means of introducing vascularization, we'll be getting vascularized brain organoids, which may be useful in developing a human brain at all, and we will be even able to transplant this into the instead of our normal brain. Right. Now coming for the applications. So applications of the brain organoids is a very, very important thing. And I told you in the recent years, it is gaining lots of importance, mainly because of the success they could find in the brain organoids. In brain organoids, it has got various applications. Number one is identifying and checking and treating the neurodevelopmental disorders, neurodegenerative disorders, mental illness, toxicology. Say, for example, how our brain responds to nicotine or any other toxin for the matter, and brain tumors, and even for identification and treatment of the infectious diseases. It is not only for identification and treatment, it is also to study, because how our body or how our organ responds to every disease or every condition differs. Right. So in 2019, they were working, and the various neurodevelopmental disorders that were treated using these kind of brain organoids include primary microcephaly, acquired microcephaly, autism spectrum disorders, periventricular heterotropia, neonatal hypoxic injury, and Down syndrome. And we also know neurodegenerative disorder, Alzheimer's. And we all know that Alzheimer's is a number one ranking neurodegenerative disorder. And what they have done is, they worked on the organoids, brain organoids, and they tried to correct that particular gene responsible for the Alzheimer's by means of CRISPR technology. And that is the thing they have done in correcting the neurodegenerative disorders. And they have also worked on Parkinson's disease and also in brain tumor. And coming for infectious diseases of the CNS, neurotropic virus infections, cerebral malaria, and in mental illness, schizophrenia. So the brain organoid have got the vast application of treating all these disorders. So next is gut organoid. So next to the brain is gut. Because gut organoid is, again, it is gaining a very big market throughout the world, and it covers almost everything, right from stomach to the oral to the colon. So basically, the gut organoid refer to the organoids that could recapitulate the structures of the GI tract. Okay, so this tract arises from the endoderm, and again it can be differentiated into three. That is, the foregut gives rise to the oral cavity and the stomach. The midgut gives rise to the small intestine and the ascending colon, and the hindgut gives rise to the rectum and the rest of the colon. And the next is the intestinal organoid. So though it comes under the gut, it is being treated separately because of the market we have got in intestinal organoid. The intestinal organoid are the gut organoids, and this is generated directly from the pluripotent stem cells, and these are also produced from human pluripotent stem cells and generated from intestinal stem cells, which are extracted from the adult tissue. This is a quite common problem which we are coming across throughout the world. So in most of the cases, we can isolate the cells from the patients, and the patient's cells can be converted into the induced PSCs, and from that we will be able to make the crypt-villus structure of the intestine. The speciality of this intestinal organoid is it is maintaining the same villi structure inside a test tube also. I mean, in your plate also. So you can find what is actually happening inside your intestine, and you can mimic the same in your laboratory. So in this case, this intestinal organoid also is considered to be a valuable model to study the intestinal nutrient transport, drug absorption, as well as hormone secretion. And this intestinal organoid is also gaining lots of value because of the high degree of fidelity it gains. Regarding the fidelity, it is always a problem again in case of organoids. Not the question of fidelity, but some of the organoids can never, or what to say, they mimic the organ, but they will not be able to, or they may not be able to reproduce the entire properties of the organ. Say, for example, if you are going to grow a kidney cell, it is not it need not necessarily show all the properties of a kidney cell. But of course, it may be showing certain important properties and you have to work on it. And since I told you this is a developing research, developing medical research, many people are working on each and every properties of every organ. They make lots of organoids, they come out with new products, which shows a different properties or functions. The intestinal organoids right. The intestinal organelles have also demonstrated therapeutic potential. Right. And next is stomach or gastric organoids. So coming for the stomach or gastric organoids, they also have got the capacity of recapitulating partly the physiology of the stomach, and this can also be grown directly from the pluripotent stem cells or even from the adult cells. So this is how it goes off. And there is a company, HUB, Crown Bio, and they developed the 3D organoid culture development in this. They got their own protocol. I told you, the leaders, the brand leaders they develop their own protocols and their own products of organoids. So this particular company is producing this way. That is, they take the embryonic stem cells or a skin fibroblast, and by means of reprogramming the skin fibroblast, they get the pluripotent stem cell culture, which on endodermal differentiation converted to spheroids, and the spheroid culture is now fixed onto the matricial culture, and now it is now getting converted into the intestinal epithelial and mesenchymal organoid. Similarly, if you are going to get the adult small intestine can be minced into small pieces, and then the minced small intestinal tissue can be converted into a spheroid, matricial culture, put into a matricial culture, and can be grown. Or else, the same thing, they isolate the small intestinal crypt, and by adding certain stem cells, here LGR5 plus stem cell line is being used, and by combining these two and growing in a matricial culture, they could make intestinal epithelial organoids. So to be simple, whichever organoid you are in need of, you can approach these kind of companies, and we can give an order, and they will be making your own organoids. Okay. So that is it. And next is lingual organoids. So lingual is nothing but tongue physiology. So this is a very important part of this is taste. So we all know taste plays a very important role in food industry, and that is how the lingual organoid plays a very important role in food industry. Initially, they produced a lingual organoid which lacked taste receptors, but recently they have also created a lingual organoid with taste receptors by means of growing a progenitor cells from the circumvallate papilla tissue. There are also other types of organoids which are available in the market. Number one is thyroid organoid, which can give you thyroid. Number two, thymic organoids, which is grown from the thymus. And this thymus have got the capacity of growing or making your T cells. So for T cell productions, the thymic organoids are in the market. And then testicular organoids, which are important for reproductive biology. So we all know that today lots of hospitals are there throughout the world, of course, we can say, mainly for the reproductive system. And this testicular organoid is gaining its own market value in that particular field. And similarly, hepatic organoid for liver, and pancreatic organoid, epithelial organoid, lung organoid is gaining importance recently after this COVID. They have started making lung organoids, and they have started studying about the lung organoids for observing how the COVID virus enters into the lungs and how it affects it. And similarly, the lung organoid is recently used for developing vaccines for COVID. And kidney organoids, this again is a separate field, and kidney organoids gains very important market value because of the necessary for kidney transplantations. Similarly, gastruloid, blastuloid, and in 2018, very importantly, even cardiac organoids have been developed, where they have started producing even cardiac organoids, and it is still under process. And again, retinal organoids are also there. So these are the various organoids which are being produced by various brand leaders throughout the world. And there are two ways by which they produce. So either by means of pluripotent stem cells or adult cells. So in case of pluripotent stem cells, either they take the blastocyst, ICM, they produce ESCs and iPSCs, and they make endoderm and ectoderm. And from endoderm, they will be able to make gastric organoid, intestinal organoid, liver organoids, thyroid organoids, and lung organoids. And from ectoderm, they'll be able to make optic cup organoids, stratified cortical organoids, pituitary organoids, and inner ear organoids. Similarly, by means of adult cells, by taking adult stem cells, we will be able to make mammary gland organoids, liver organoids, pancreas, gut organoids, prostate organoids, stomach, lung, as well as tongue organoids. So this is how we can make various organoids. And we are into the last part, that is not last but next is applications of organoids. So coming for the applications, the various applications of the organoids only make these organoids as a very important field. And I told you, it is a $500 million business, right? So coming for growing business, it's a growing business. So coming for the various applications, number one, it is used for studying the development and disease modeling, precision modeling, regenerative medicine, toxicology, drug discovery, host to microbe interactions, gene editing using CRISPR technology, omics, phylogenetic studies. So these are the various applications. Just we will see very few. Number one, how it is used in basic research. So in basic research is nothing but understanding the biological processes. So organoids are able to show how the cells interact in an organ. Can you hear?
Now, a small disturbance like here and there.
Okay, shall I continue?
Yeah, you can continue. You can continue.
Okay.
Now
So in basic research, the organoids are the excellent tool to study the basic biological process. That is to understand what is actually happening, how the organs respond, how the organs work, and how it takes the nutrients, how the drug is transported, how the hormones are secreted. All those stuff can be explained by means of these organoids. And similarly, for understanding the metabolic diseases, mainly for studying metabolic diseases, insulin resistance, diabetes, all the stuff it has become very easy to understand because of these organoids. Number two is models. It can function as a good model for diseases. For various diseases, we'll be, instead of taking the tissue as such and working on it, we can induce a disease into an organoid and see what and all changes it is undergoing. So this is possible in case of using organoids. And recently they have developed, they studied polycystic kidney diseases and focal segmented glomerulosclerosis using the kidney organoids. And in developmental biology, it is also functioning as a model for developmental biology. So basically, throughout the European countries, mainly in European countries, most of the research works are going on the developmental biology and experimental processes of understanding molecular clock and organogenesis. Because understanding the basic can save you a lot of time in your applied research. So in this case, say for example, brain organoids are very well useful in studying the development of the central nervous system. Similarly, for the development of any organs, we can go for these kind of organoids. And fourth is personalized medicine. There is a company called Clever's Group. They were the first one to produce the personalized medicine. They produced intestinal organoids from the rectal biopsies of the patient, and they could develop a drug for not exactly drug. They could make organoid as a treatment for cystic fibrosis in that particular patient. So what they could do is they could encode a gene for epithelial ion channel, which is necessary for a healthy epithelial surface fluid, and they could replace that with the newly developed organoid. So this again is another example how to generate a pancreatic beta cells for the treatment of diabetes. Say for example, this is a quite common example. We know that many of the people have got the capacity of, they are getting affected with diabetes, and when their pancreatic beta cells are in a problem, we can go for this kind of techniques where the HESC adherent culture is grown and it is converted to pluripotent cells. We can harvest it, and these single cells will be mixed with HUVEC cells, that is human umbilical vein endothelial cells, and these will be grown in an amica gel, and this can be converted into an organoid which makes islets. Right. If not, you can select a patient, and from that particular patient, you can isolate the islet, and even with the very small amount of cells, we will be able to co-culture it. Again, we are going to combine with the HUVEC, and here we can also introduce the vascularization to get a vascularized islet-like organoids, which will be able to produce your beta cells. So this is how it is used. And again, the next one is drug discovery. So the biggest problem in drug discovery industry is the cytotoxicity studies. So this is the important thing which makes a drug potential. Right. Now, for cytotoxicity studies, we are basically dependent upon the animal models or cell models. So instead of animal and cell models, we can go for this kind of organoids, so that this can give you the fidelity, this can give you the relevance of the drug functionality as well as the toxicity of the humans, toxicity for the humans with high fidelity. And similarly, by means of using liver organoids, we'll be able to exactly decipher. We will be exactly able to decipher how a liver is responding towards a drug. So the drug metabolism can be deciphered using this. Similarly, the next application is regenerative medicine. We all know, I told you already, there are many cases where the organoids can be used as it can be used as a regenerative medicine. That is, say for example, in liver failure, we can use these liver cells, liver organoids, and in near future, it is always possible to replace the organ transplantation using these organoids, and it is also useful for correcting genes. Say for example, if a person has got a tissue with a mutant gene, it is possible for us to take away that, correct it using CRISPR technology, and then grow into an organoid, and it can be kept back to the, or transplanted back into the person. And last important application is deciding which culture conditions to use. So basically, this is of industry related point, because in every industry, coming out with the procedure is very, very important, and different kinds of organoids are grown in different culture conditions. So developing a matrix or developing a culture condition is very, very important in terms of money. So in these cases, the chemically defined hydrogels or any other natural or synthetic hydrogels or matrices can be tested using these kind of organoids. And these kind of organoids help in deciding the 3D culture with the supportive matrix or suspension culture or air liquid cell pellet culture. See? This is how it is basically grown. So 3D culture in supportive matrices, we can grow intestinal organoids and gastric organoids using natural ECMs. And similarly, intestinal organoids can be grown in synthetic matrices also. And cerebellar, that is brain organoids, can be grown without ECM proteins in 3D suspension culture. And optic cup organoids can be grown in 3D suspension culture with ECM proteins. And kidney organoids are grown in air liquid cell pellets. And last is genetic engineering, as it is nothing but, as I told you, the CRISPR technology, we can do gene repairs. So we are into the last part, that is the market scope. So coming for the market scope, in 2019, the value was 405 million, and in 2020, it is $500 million. And see the growth rate, it is 22%. And basically, North America is the leader in this, sharing somewhere around 41% of the entire share of the whole world. And out of all this, the biotechnology and pharmaceutical industries gain a biggest share, followed by the academic and research institutes, and then only hospitals and diagnostic centers. Here, the second, that is academic and research institutes, are gaining a bigger market because it is still under research. And coming for the international market, as I told you, US and Canada biotechnology sector have already taken this organoid as a very important sector, and they are growing. And in Asia Pacific, China is leading. And these are the various key companies, that is 3D Biometrics, HUB, Lonza, Thermo Fisher, Greiner, etc. These are some of the few important companies which compete among each other, and they often come out with all new improved products. Say, for example, very recently, PerkinElmer have come out with a simplified version of a detection, handling, and analysis of the spheroids. And similarly, some of the companies have developed mini lungs. I told you lungs. So mini lungs they have developed for the SARS-CoV, for understanding the mechanism of SARS in the lungs. And like any other market, COVID also has got its own impact on the human organoids market, because throughout the world it has stopped the manufacturing and delivering of the processes, and similarly it has disrupted the supply chain. And more than all, there was a bigger lockdown throughout the world, particularly in countries like US, France, and UK, where we could find a large amount of COVID infection. Everywhere, the organ donation and transplantation business have gone down. And this ultimately affected this organoids also. And this is the prediction in India. So in India, in 2016, the market was $7.4 million, and in 2017, it was 9.2, and there is a prediction that it will be crossing $1 billion in 2027. So coming for a conclusion, so thus the recent development of a 3D stem cell derived organoid culture systems have allowed a generation and long-term expansion of the adult tissues far beyond the predicted limit. And similarly, studying the molecular mechanisms involved in certain diseases, finding solutions in terms of therapy, and even correcting the genes and transplanting them have made these organoids as an important option in the recent medical research. So these are few references, and thank you.
Thank you, Murali sir. It was an excellent session. So we gone back into nook and corners of all the research part and topic regarding organoids. So over to you, Kavya. So the participants, if there is any queries, you can post in the chat box. Or else you can unmute and you can ask.
Actually, this one particular topic is going to lead everyone. Of course, in future, we are going to have our own laboratory where we have to work. We are going to have what to say. We are supposed to work on these organoids in future. We will be replacing our own laboratories. Because no more, we are not going to have any more animal models, and this is going to lead entire research actually. So any questions?
So there is an appreciation, and there is a question from Devika ma'am. What are the application of brain organoids?
I told you, brain organoids can be used in various ways for identification and treatment of various neurodegenerative diseases. We'll be able to identify the drugs for neurodegenerative diseases. We can replace certain brain parts using these brain organoids, etc.
So what are mini brains? One more question has been posted. What are mini brains?
Mini brains are nothing but miniaturized brains. It is not exactly brain, but it can function like a brain. By taking the brain cells, we are developing a small brain which can function like a brain. So instead of giving a drug to our brain and seeing what is happening, we cannot do it ethically. Whereas we can make a brain, a miniature brain, and we can give a drug to it, and we can check what is happening. So this will be very much useful in. That's the thing which we are doing. Actually, we are taking animal models. We take an animal model, we give it something, whatever you want to test, you give it, and then you dissect it, and you take the organs, and you are going for the study. Instead of that, you can go for an organoid. You can go to these companies, order for an organoid, and you select an organoid, give your own drug of interest, and then you can check. You can check what actually will be happening inside a human brain. You'll be able to see. That's what is called as a mini brain.
Hope Devika ma'am, you got the point. So there are many appreciation messages, like the session was excellent, and thank you so much for the nice presentation with that. So still we are getting messages from the personal WhatsApp messages and the group also, sir. So thank you so much for the excellent presentation. Sir, can you share your email ID over here in the chat box?
Can I share?
Yes, sir, you can.
Email ID.
Yeah, your email ID.
VMK Biochem.
Yeah. Shall I type for you?
Okay, shall I message it?
Yeah, you can message it in the chat box.
Okay, right. Yeah. So can we end up, ma'am?
Yeah, 1 second, sir. So over to you, Kavya.
Yeah. Thank you, sir. Thank you for sharing your valuable knowledge with us. Now, I would like to call upon Ms. Anita, Assistant Professor, Department of Biochemistry, to give a vote of thanks. I welcome you, ma'am.
Thank you, Kavya. Good afternoon, all. It's a great honor and privilege to propose the vote of thanks on this memorable session. First and foremost, I thank our guest, Dr. V. Muralikrishnan, Assistant Professor, who spared time from his busy schedule to grace the present occasion. The present session was full of knowledge and interesting. It gave deep insights into the topic. Thank you, sir. I take this opportunity to express our sincere thanks to our Principal Sir, Dr. T. Shivkumar, Vice Principal, Dr. Ayapada Sir, and DBT Staff Coordinator, Dr. Dhanabalan Sir, for the moral guidance. I would also like to thank our beloved HOD, Ms. Shamina Ma'am, for her valuable support and guidance. Finally, I thank participants for your cooperation to make this session a resounding success. Thank you all.
So thank you, Anita ma'am. So once again, on behalf of the management, principal and the vice principal and all DPT college scheme, we heartfelt thanks to Murli sir. So we feel blessed with the thoughts, the things that you really shared. And I saw so simplicity in you, like when you were asking, I wanted to be an attendee to this program, and you are also into our WhatsApp group. Really, it's so like it's a blessing over here. So thank you so much, sir. You should also always be connected with us to share more thoughts about how to shape biochemistry in different angles of research. So really, we need your guidance, sir. So stay connected with us. So like in the next academic, like when the next session starts, really we will call you again for this any type of DPT sponsored programs, and you have to be with us for the program. Thank you so much.
Sure. Thank you so much.
Yeah.
Thank you for giving me an opportunity.
Thank you, sir. So all these sessions will be are video recorded, as you know that. So this will be converted to your link, and the link will be shared in the WhatsApp group. Okay. So probably tonight I'll be sharing the last 4 sessions, what has happened yesterday and today. So on daily basis, the forenoon session and the afternoon session video links will be shared with you. Okay. So you can also watch through it. You can also share it and other things. So thank you so much, all the faculty members. Thank you, sir. So for all the faculty members, and we have Mr. Narayan sir with us. Sir, do you remember Narayan sir?
Yes.
He was one of our faculty. He must, I think he's
yes.
yeah, he's now connected with us. So like Manju ma'am, you want to share any words, few words with us? So they are on the mute stage. Okay, sir, no problem. So we will meet again in the next session. Thank you. Before you leave the session, kindly look into the video links that are, feedback links that are being displayed in the chat box. Complete your thing, because that will help us to scrutinize and regularize the certificate's purpose.
Thank you. Thank you. Thank you.
Okay, thank you, sir.